MCAT Biochemistry Topic 1 Amino Acids. There are 5 important topics to discuss when it comes to the amino acids on the MCAT. That is 1. The classifications of the amino acids. 2. Peptide linkage and hydrolysis. 3. The resulting protein structure caused by the peptide linkages. 4. The acid-base properties of amino acids. And 5. The stereochemistry. To begin the discussion, it's important to realize that when talking about amino acids in the context of medicine, and specifically the MCAT test, we are talking about the 20 proteinogenic amino acids. These are called proteinogenic because they generate genic proteins. So there are other amino acids that you can synthesize, and many will be used for other things. But when it comes to nature, either whether it comes from food or made in the body, there are only 20 that are actually put into making proteins. So proteinogenic amino acids are the topic of this discussion and what you will encounter in the MCAT and medicine generally. All right, so the first important thing to discuss is topic number 1, which is categories of amino acids. So the amino acids can be broken down into 2 general categories, and then further broken down into 2 more categories within those 2 categories. So what do I mean by that? The first 2 categories are hydrophobic versus hydrophilic. The categories within those are, for hydrophobic, we have aliphatic and aromatic, and for hydrophilic, we have polar but uncharged and polar but charged. So we are going to go through each of these categories, and in doing so, elucidate the full name, 3-letter abbreviation, 1-letter abbreviation, and structure and special qualities of all the amino acids. And for the reference to the source information for this, we have Kaplan's MCAT Quicksheets, we have the Miles Down study guide for amino acids, and we have notes from the Leah Forsyth video about amino acids. Okay, so first let's start with hydrophobic amino acids. So hydrophobic means water-fearing, and in the context of amino acids, this means not soluble in water. So just to recap about what soluble versus not soluble means, when you have a solute that you dissolve in a solvent, the extent to which that solute will dissolve in the solvent is determined by the similarity in chemical structure between the solute and the solvent it is dissolved in. And since the chemical structure of water is that of a very small and polar molecule, with the oxygen-hydrogen bond being very polar, you need a small and very polar solute to dissolve in water. So bringing it back to hydrophobic versus hydrophilic, the hydrophobic amino acids will have R groups that readily that do not dissolve in water, and the hydrophilic amino acids will have R groups that do dissolve in water. So they interact favorably with water, whereas the hydrophobic ones do not interact favorably with water. It's really not much about dissolving here, as it might have been in general chemistry's discussion of solubility, but it's more about the interactions that these amino acids will have within the proteins that they form. All right, so when it comes to non-polar hydrophobic amino acids, hydrophobic and non-polar are pretty much synonymous in this case. We have 2 main categories: aliphatic and aromatic. Aliphatic means saturated with hydrogens. Aromatic means containing an aromatic ring. So there are 9 total hydrophobic amino acids, and 6 are in the aliphatic category, and 3 are in the aromatic category. And one of those 3, it is important to mention as sort of a exception / both thing. Okay, so starting off with the aliphatic, we have glycine. 3-letter abbreviation, GLY. 1-letter abbreviation, G. This is the amino acid containing the R group hydrogen. It is the only achiral amino acid. It is usually found inside the protein because it is hydrophobic. So it is on the inside of the protein, inside hydrophobic, outside hydrophilic, because the exterior environment with which the protein will be interacting is water. So you'll want the hydrophilic ones interacting with water on the outside, and the hydrophobic ones staying away from water on the inside. And glycine is one of those inside amino acids. So I realize now that I neglected to describe the structure of the amino acid backbone, so I'll do that now and then explain a little bit of why the R group is the only thing that I am talking about when I mention each of these individual structures. Okay, so the prototypical amino acid, it contains one central sp3 hybridized carbon bound to, on the right side, well, it's really not right or left, but let's just say on the right side you have a carboxylic acid, and then on the left side you have a primary amine. So the central carbon is what we call the alpha carbon, and it is bound to the carbonyl, which is then bound to the OH of the carboxylic acid, and then on the other side it is bound to the NH2 of the amine group. And we know that at physiological pH, you have a pH of about 7.3-ish, 3.5, I think. So at physiological pH, a carboxylic acid is, if it's 7.5 and the pKa of carboxylic acid is 2, then it will be deprotonated. So you will have a negative charge on your carboxylate terminus, and then the opposite is true for the amine, so that will be protonated at physiological pH. And I'm going to get into more of the acid-base chemistry later on, but basically, yeah, NH2 protonated becomes NH3, carboxylate deprotonated becomes COO minus, and so when you have a positive and a negative, if you sum the two charges there, you have neutral. And this is the Zwitter ion form, which is what you find at physiological pH for your amino acids. But why am I only mentioning the structure of the R group here? Because, in fact, the backbone, which is that carboxylic acid and amine, the backbone is a neutral chain of those structures, and so you actually don't usually have the amine and the carboxylate just hanging off there interacting with the surrounding environment. Really, you just have the R group, and then that whole backbone is like a long chain where everything is just being held in place, and we're not going to talk about the acid-base chemistry specifically of that until we get later on into the finer details of the acid-base chemistry. Okay, so back to glycine, number 1 amino acid, aliphatic, hydrophobic. Glycine is also found in the twists and bends of the protein structure, which could be an alpha helix, for example. So this is a 2' structure, secondary structure, alpha helix. Has twists and turns and bends, and you might find glycines in the kinks of those bends because it is so small. Second one is alanine. The R group of alanine is one methyl group, which is a CH3 group. Alanines are hydrophobic because that CH3 group does not interact favorably with water. Methane is not soluble in water, neither is alanine. Alanine's R group. A very important thing to mention about alanine is its metabolic properties. So if you take an alanine and you react it through a reaction with this molecule named alpha ketoglutarate, which is a alpha keto carboxylic acid with an ethyl group and then another carboxylic acid at the end, through this reaction called transamination, you create pyruvate from your alanine. let me just describe what this reaction looks like. Okay, so you have alanine. Let's say that alanine has its NH3 group on the left and its O minus group on the right, and then a CH3 coming off. Well, we're going to take that NH3 group, remove it, and replace it with a carboxy group, carbonyl group. So you take off the NH3, you replace it with the carbonyl. That is actually pyruvate. So the structure of pyruvate is CH3, carbonyl, ketone, and then carboxylic acid. So and then in creating that, you need to give the NH3 group to something, and you actually give that NH3 group to a molecule called alpha ketoglutarate, which is a 1, 2, 3, 4, 5 carbon chain with carboxylic acids on either end, and one of the carboxylic acids has an alpha ketone. So that alpha ketone pops off, gets given to alanine, and the NH3 group from alanine replaces that ketone group on alpha ketoglutarate. And keeping track of all that together, alpha ketoglutarate is then converted to glutamic acid by the addition of the NH3 group. So the result of transamination of alanine is 1 pyruvate, 1 glutamic acid, and the reagents for that are alanine and alpha ketoglutarate. That is an important quality of an alanine to mention here. The MCAT Quicksheets does not mention that, but I would certainly remember that. Okay, so alanine's 3-letter is ALA, alanine's 1-letter is A. Next, valine. Valine's 3-letter is VAL, valine's 1-letter is V, and its R group structure is 1 isopropyl group. This isopropyl group is bulky, and therefore valine is not found in the turns of alpha helices like glycine is. Okay, and it is hydrophobic. This is all the hydrophobic section. Next is leucine, LEU, L, and isoleucine, ILE, I. An important note here is that the 3-letter abbreviation for isoleucine is not ISO, it is in fact ILE, and the 1-letter is I. Okay. Really no important properties to mention here, except for the fact that if you think about it, isoleucine not only has a chiral alpha carbon, which is common to all amino acids except for glycine, but it also has a chiral center in its R group, in its side chain. So that first R group, well, let me first describe the structure, actually. I realized I forgot to do that. So leucine structure is the same as valine, except you add a CH2 group between the alpha carbon and the isopropyl group. Isoleucine structure is the same as leucine's, except you take one of the methyls, the CH3s from the end, and you stick it onto that CH2 group that comes right off the alpha carbon. So you have alpha carbon, CH bound to, CH bound to CH3 and CH2, and then that last CH2 is bound to another CH3. You do sort of have to see this in person, but the reason it's called isoleucine is because it is a constitutional isomer of leucine. Number 6, methionine. Methionine's 3-letter abbreviation is MET, M-E-T. Methionine's 1-letter abbreviation is M. Methionine is the amino acid with the R group that has a CH2, a CH2, a sulfur, and a CH3. So 1, 2, sulfur, CH3. And you may think, given that you have a sulfur atom, that this R group would be polar. Not the case, because the difference in electronegativity between sulfur and carbon is not strong enough to make this a polar amino acid. It is very important to note that methionine is the start amino acid, meaning you, so you basically, it started, like every beginning of the genetic code will start with methionine. And the codon for methionine is AUG. Just look that up real quick. Okay. Number 8, phenylalanine. So we have now gotten into our aromatic hydrophobic amino acids. The first is phenylalanine. So phenylalanine is a glycine where you add a benzyl substituent. The benzyl group is a CH2 plus a benzene ring. So that is phenolalanin's R group. You have a CH2 and a benzene ring. Really no important properties that Leah mentioned here, but one, I suppose you could say, is that it is aromatic. Aromatic, can do certain things. I'm not exactly sure, but yeah, benzyl substituent on phenylalanine, nonpolar, hydrophobic. There's no polar bonds here, no difference in electronegativity here. So nonpolar, hydrophobic. Okay, phenylalanine, the 3-letter is PHE, the 1-letter is F. And that is because the P is already taken by proline, which we will mention soon. Okay, another amino acid that is hydrophobic, and the final one here is tryptophan. Tryptophan's 3-letter abbreviation is TRY, and tryptophan's 1-letter abbreviation is W, tryptophan. It is the only bicyclo amino acid, meaning that it has 2 rings, and it is not polar, despite the presence of a nitrogen in its backbone. So let me just quickly describe the structure of the amino acid tryptophan. You have the alpha carbon double bound to a carbon, which is single bound to an NH, and that NH then ends. That alpha carbon is also bound, that NH then is bound to a carbon. The alpha carbon is also bound to a single bound to another carbon. Now, the NH carbon and the alpha carbon's carbon are bound to the 2 carbons on a benzene ring. So, you have 2 rings. One is a 5-membered ring composed of a carbon-carbon double bond, a carbon-nitrogen single bond, another carbon-nitrogen single bond, a carbon-carbon double bond from the other benzene ring, and then a carbon-carbon single bond to the alpha carbon. You'll notice all of these atoms are sp2 hybridized because you have lone pairs or double bonds or a lone pair bound to a double bond, which is also sp2 hybridized. Okay. The final non-polar amino acid to mention is proline. Proline is not necessarily aromatic. I'm not sure why it was included in that category, but proline's structure is unique among the amino acids because the nitrogen on its backbone is part of the R group. It is bound to the R group. So your alpha carbon is going to be involved in a 5-membered alkyl ring that includes the nitrogen of the amine group. So you have a CH2, a CH2, a CH2, and your N, and then the alpha carbon again. So it's a 5-membered ring with your amine group and some alkyl carbons, and then of course you have the carboxy group of the amino acid flying off the other end. Now, what is helpful about this structure, remember the amine nitrogen of an amino acid is involved in the peptide bonds between the amino acids, meaning that when you want to form a kink in your alpha helix structure, you use a proline, because that proline's backbone will necessarily be sp3 hybridized and form a turn in your structure. So it is not linear in structure like the peptide backbones of all the other amino acids when they make peptide bonds. Instead, it is going to be sort of like kinky. It is the kinky amino acid. Okay, so proline's 3-letter abbreviation is pro, and its 1-letter abbreviation is P. And important properties to mention, okay, it is the only cis amino acid. The side chain is part of the peptide bond, yes, and introduces kinks and alpha helices, yes. I'm just reading off of the Kaplan QuickBooks right now, and then it is found in loops and turns. Okay, we all know this about proline. Now let's turn to the hydrophilic amino acids. And within the hydrophilic amino acids, there are 2 categories. One is polar, meaning that it contains covalent bonds with a significant difference in electronegativity, and one is charged, which means beyond just being polar, you then have a charge on one of your atoms. And that'll be more clear when I go in to describe each amino acid. Okay, so there are 11 here. You'll remember that I mentioned 9 before, so now there are going to be 11, and I'm going to go through them. Oh, okay, so number 1 of the polar amino acids, the hydrophilic amino acids, is serine, S-E-R-I-N-E. Serine's 3-letter abbreviation is S-E-R, and its 1-letter abbreviation is S. This is an amino acid that contains a CH2. As its R group, it contains a CH2 and an OH. So it's literally just a methanol coming off of the alpha carbon, if you want to think of it that way. One important property that is bestowed upon serine by that OH group is its ability to participate in hydrogen bonds. The OH functional group is a hydrogen bond donor and a hydrogen bond acceptor. So it makes very good hydrogen bonds with water, which makes it incredibly hydrophilic. You'd probably find this on the outer side and in like the functional part of a protein. Okay. Another thing that hydroxyl group can do is this thing called phosphorylation, where you remove the hydroxy oxygen, or you remove the hydroxy hydrogen, and you replace that with a phosphate group, which is PO4 3-, and then that becomes negatively charged. Okay, so serine, phosphorylation, hydrogen bond donor. Another thing that Leah thought important to mention is that it can participate in reversible binding, which is very important when you have an active site in a protein or an enzyme, etc., and etc. And so those are all properties of serine. Very popular, very important amino acid. Okay. Number 2, threonine. The 3-letter abbreviation for threonine is THR. The 1-letter abbreviation for threonine is T. Threonine has the same R group as serine, except that the carbon bound to the hydroxyl is also bound to a methyl group, i.e. a CH3 group. The threonine's hydroxy oxygen is also capable of reversible binding in the active site of a protein and phosphorylation, which would turn it into an OPO3, 2- group. Okay, just like with serine. All right. So, however, that methyl group does bestow another quality onto threonine, which is that it is not good for forming alpha helices. So, you're not going to find threonine in your kinks in the alpha helix of the 2' structure of the protein because it has that bulky methyl group coming off of your hydroxyl carbon. Okay. Number 3 is cysteine. Cysteine. C-Y-S-T-E-I-N-E. The 3-letter abbreviation for cysteine is CYS, and the 1-letter abbreviation for cysteine is C. Now, cysteine is a amino acid with the structure CH2 S bound to H. That's the structure of the R group. Of course, it has the backbone, but that's the R group. CH2 S H, which makes it a thiol group. Now, some may say that cysteine is hydrophobic. Some might say that it is hydrophilic. And the difference in this opinion comes from the fact that the SH bond is not as polar as the SO bond, or the HO bond, rather. So it could be determined to be nonpolar, it could be determined to be polar, but its hydrophilicity is not the property that we would focus on when discussing cysteine. A very important property of cysteine is its ability to drop the E and form cysteine. And what does that even mean? Well, cysteine without an E, C-Y-S-T-I-N-E, actually indicates a bond between the sulfur of one cysteine and the sulfur of another cysteine. And it turns out that you can use oxidizing agents to remove the H's, right? Removing H is oxidizing. Remove the H's of both these SH groups, bond them together, and form what you call cysteine. It is also called a disulfide bridge. Di meaning two, and sulfide meaning sulfur. Disulfide bridges are the only covalent bonds that contribute to the 3' and 4' structures of proteins. I repeat, disulfide bridges are the only covalent bonds, not intermolecular forces, not hydrogen bonds, none of that, covalent bonds that contribute to the 3' and 4' structure of a protein. That is cysteine. Okay. Now we have tyrosine. That's number 4. So, tyrosine's 3-letter abbreviation is TYR, and its 1-letter abbreviation is Y. Now, it's also sort of on this hydrophilic middle ground. Interestingly, Khan Academy lists tyrosine as hydrophilic, and it also lists cysteine as hydrophilic, and given the affiliation between Khan Academy and the American Association of Medical Colleges, it might be better to associate tyrosine and cysteine with hydrophilicity rather than hydrophobicity, as Leoforside does. But regardless, that is not the most important property, right? So, tyrosine structure is CH2, benzene ring, OH. And the benzene ring is para-substituted, so the benzene ring has the CH2 on one end and the OH on the other end. They are exactly opposite to each other, just like any para-substituted aromatic ring. Tyrosine's important properties are, one, that it can participate in reversible binding, just like threonine, just like serine, and two, that it is phosphorylatable, meaning that OH group can undergo phosphorylation to gain a negative charge. So that is just like threonine, just like serine, and that is owing to the presence of the OH group. Okay, number 5 and 6 were mentioned by Leah together for their similarity in their structure. And why don't I describe that structure? So, number 5 is asparagine, number 6 is glutamine. So, these are the amide versions of aspartic acid and glutamic acid, which we'll mention later because those are also amino acids. But let's start with asparagine and glutamine first. Now, the 3-letter abbreviation for asparagine is kind of interesting. It is ASN, like an assassin. The 1-letter abbreviation takes the N from the 3-letter, and it is N. For glutamine, it is GLN, so sort of similar to asparagine there. But the 1-letter abbreviation for glutamine is Q. I question why glutamine has that 1-letter abbreviation. The structure for asparagine, R group, is a CH2 and then an amide. The structure for glutamine's R group is a CH2 and then another CH2 and then an amide. Interestingly, the letter A comes before the letter G in the alphabet, meaning that comes before asparagine has one less CH2 group between the alpha carbon and the amide than glutamine. Interesting. Okay. So let me get into the properties of the asparagine and glutamine functional group, which is basically the same thing, right? It's an amide, which is a carbonyl bound to an NH2. Now, when you have a carbonyl bound to an NH2, it is not basic. Usually, if you see NH2, it is basic, but when it's bound to a carbonyl, that lone pair is busy participating in resonance with the carbony oxygen, and it is not ready to go out and attack a new proton. So you're not going to really see that NH2 amide group forming any sort of, it's not going to get a positive charge, usually. It's really not that basic. Okay, but it can form hydrogen bonds because those hydrogens are still bound to a nitrogen, and that nitrogen sure does have resonance with the oxygen. So it is, can definitely form hydrogen bonds. And since we are now talking again about nitrogen-containing R groups, right? We discussed that before when we were discussing the amino acid. Well, we actually haven't discussed nitrogen-containing R groups. Well, tryptophan has one, but really the amino acid alanine is the one I want to take us back to, which is when we discussed transamination, where we removed the NH3 group, on the backbone, and replaced it with a carbonyl group to form pyruvate. Well, when it comes to asparagine and glutamine, you can also do a transamination reaction. But in order to do that, you would first convert the NH2 group to an OH group. The NH2 group on the amide. When you convert that to an OH, it becomes a carboxylic acid group. And that is the same structure as aspartic acid, which is the next two amino acids we're going to mention. And then through transamination, removal of aspartic acid's backbone amine, right? So you remove that second nitrogen from aspartic acid. It no longer has any amines. It no longer has any nitrogens, which means it can be a metabolic intermediate because, metabolism, we don't want really like toxic nitrogens around. And the name of that metabolic intermediate is oxaloacetate. acetate. Okay, and if you do the same thing to glutamine, all we have there is an extra CH2 between the alpha carbon and the functional group, the amide. So we remove the NH2, replace it with an OH, and that makes glutamate. And then we remove the NH2 of the backbone, that makes alpha-ketoglutarate. You'll remember alpha-ketoglutarate from my discussion of alanine. It is a 5-carbon long metabolic intermediate that has 2 carboxyl groups on either end and then an alpha ketone on one of them. Alpha-ketoglutarate. Okay, so asparagine can form oxoalloacetate, glutamine can form alpha-ketoglutarate. Those are important metabolic intermediates, which makes the fact that they can form those important qualities of asparagine and glutamine and aspartic acid and glutamic acid. Okay, so now let's get a little bit deeper into aspartic acid and glutamic acid. These are the 2 acidic amino acids, and they are such because they have carboxylic acids functional groups. Now, the 3-letter abbreviation for aspartate or aspartic acid is ASP, and the 1-letter abbreviation is D, aspartate. The 3-letter abbreviation for glutamate or glutamic acid is GLU, and the 1-letter abbreviation is E. You can think of this in a similar alphabetical way as we did with asparagine and glutamine. Aspartate, coming before glutamate, has D, and then right after in the alphabet is E. Alright, so given that these are the carboxylic acid analogs to asparagine and glutamine, their structures can be described similarly, and being very hydrophilic because a carboxylic acid group forms and accepts hydrogen bonds, they will be on the outside of the amino acid. So you'll find these guys on the outside of the amino acid. An important thing to mention about these acidic amino acids is that they will form what we call salt bridges. So, referring now to the Biochemistry MCAT Quicksheet of Kaplan, they mentioned for both aspartate and glutamate that they form what is called salt bridges. And salt bridges are part of the 2' structure of the amino acid. So, the 2' structure is... Oh wait, okay, no, sorry. Actually, I meant the 3' structure. So, you have hydrophobic interactions. Okay, I'm reading from the Kaplan book right now, or the Miles Down MCAT Quicksheets. 3' protein structure is the 3D shape of the single polypeptide chain, and it is stabilized by hydrophobic interactions, acid-base interactions, H bonds, and disulfide bonds. So, the only covalent bonds between the elements of the 3' and 4' structure, as I mentioned before when talking about cysteine, is the disulfide bond. However, the acid base, the E in base could be the 3 and 3', is another interaction that occurs on the 3' level. And when we're talking about acid-base interactions, well, you need an acid and you need a base. So we'll need those acidic amino acids, being aspartate and glutamate, or aspartic acid and glutamic acid, and we'll need the basic acidic acids in order to do those 3' salt bridge interactions in your protein structure. Now, there are 3 basic amino acids that are important to mention. The last one is very important. Okay, so one is lysine. The 3-letter abbreviation for lysine is LYS, as one might expect. However, the 1-letter abbreviation is a little bit unexpected, which is K. The structure of lysine's R group is a butyl alkyl chain, 1, 2, 3, 4 carbons, and then an amine group, primary amine. Now, an amine group is a basic functional group, meaning that lysine forms salt bridges with aspartic acid and glutamic acid on the 3' structure of the protein. So, at physiological pH, that NH3 group is going to be protonated, and it is then going to be able to donate or receive that proton from, let's say, glutamic acid, carboxylic acid. All right. And where are you going to find a lysine? Probably external, because that NH3 group is pretty polar, and it's also charged, which means it is very hydrophilic, hydro-interacting. Okay, the second basic amino acid is arginine. I say it that way because arginine's 3-letter abbreviation is ARG, and its 1-letter abbreviation is R, like a pirate. Okay, so arginine's structure is 2 CHG groups, and then this very interesting structure there at the end that I sort of just need to, I need to mention one more thing about lysine. Kaplan mentions that not only can its side chain form a salt bridge, but it can also be acetylated to mask the positive charge, which is important in DNA-protein interaction. So I'm guessing what happens here is that the amine is converted into a secondary amine, which I guess is less basic for some reason, and then you mask the positive charge. I'm not exactly sure how that works, but I suppose when we get into DNA, that'll become relevant. All right, so let's talk about arginine. Arginine, ARGR. Its structure is CH2, N with H, and then C double bound to N with H, and then, which is an imine, and then an N with two H's. So it's three N's bound to the same carbon, and then a CH2 group bound to one of those N's. You really do need to draw this one out in order to see it. Well, this is mentioned as a basic amino acid, but which nitrogen is the basic one? The basic nitrogen is going to be the one whose lone pair is not involved in resonance, and it turns out that that is the nitrogen on the double bond to carbon, because the double bond is the lone pair involved in is the electron involved in resonance, and then the lone pair on the NH group coming off the double bond with carbon is the one that can go out and grab a proton, which would make it an NHH double bound carbon. Okay, so yeah, that is a salt-bridgeable amino acid, arginine. Okay, and we'll probably get into its pKa later on. Now, histidine is the last amino acid to mention, and its pKa is important to memorize. It has a pKa of 6.5-ish, I guess. So histidine's three-letter abbreviation is HIS, its one-letter abbreviation is H, and its structure is a CH2 group, and then a five-membered ring comprised of a desaturated carbon-carbon bond, and then two nitrogens with a desaturated carbon in the middle. So one of those nitrogens is double bound to that carbon in the middle, one of them is not double bound to that carbon, and thus has an H coming off of it. Now that H is the proton in question, so when that group is an NH2+, because it's the secondary nitrogen, it can undergo acid-base reactions. So why is histidine so important? It is important because it forms interactions, it is a proton shuttle for the rest of the protein, okay? So its ability to exchange that proton, given its pretty close pKa to physiological pH, makes it a pretty good shuttle of proteins. Now, speaking of proteins, how do we form proteins? Well, we need to do this thing called peptide linkage, which is just simply combining amino acids together to form long chains of amino acids. And there are two reactions that are important to talk about when it comes to peptide linkage. One is the peptide linkage reaction, and the other is the peptide hydrolysis reaction. And I'm basically going right off of the Leia video here. Okay, so what happens in peptide bond formation is a dehydration reaction. So you're going to take your NH2 group of the amine side of the carboxylic acid, of the amino acid, and you're going to take the O from the carboxylic acid, and you are going to basically remove H2O from this whole system and form a new bond between the carbonyl carbon of one amino acid and the amine nitrogen of the other amino acid. So the amine nitrogen in the Zwitter ion form has 3 bonds to hydrogen, which makes it NH3, and also in the Zwitter ion form, the carboxyl oxygen has a negative charge. So you take the O negative from carboxyl oxygen, you take 2 hydrogens from NH3, and what you form is N bound to H bound to the carbonyl carbon, and then bound to the alpha carbon of its own amino acid. So this is a dehydration reaction because the net loss of H2O is what allows you to form the bonds. And the location of these peptide linkage formations in the cell is the ribosome, which means the enzyme which catalyzes this reaction, because it's kind of a weird reaction, you wouldn't really expect it, is called ribozyme. It's the ribosome enzyme, ribozyme. And fun fact, this enzyme is a molecule of RNA. Okay, so the peptide linkages form amide groups because the carbonyl carbon of the carboxy end combines with the amine nitrogen of the amine group, and so you have double bound O and single bound N, which is an amide group. Amides are sp2 hybridized. So in a peptide chain, the only sp3 hybridized carbons are those R group carbons, which are bound to the nitrogen, bound to the carbonyl carbon, and then bound to whatever hydrogen, and then bound to the R group, which is a hydrogen if we're talking alanine, glycine rather. Okay, so the oxygen comes from the carbonyl carbon, and the hydrogens come from the nitrogen. Now, when you form peptide chains, you can name them based on the number of carbons. So the number of amino acids that you combine to form them. So if you combine 2, that is a dipeptide. If you combine 3, that is a tripeptide. If you combine more than 3 but less than 20, it is an oligopeptide. And if you combine more than 20 rather, it is a polypeptide. Okay, so poly is designated just for those that are more than 20. So you wouldn't call something with 3 peptides a polypeptide. Okay, so what is the opposite of the dehydration reaction that forms peptide bonds? It would be a hydration reaction that removes those peptide bonds. The enzyme that catalyzes this reaction is called protease. It is a proteolytic enzyme, literally meaning it lyses proteins. Okay, so what occurs here is the exact opposite of the forward reaction, which I just described. So the nitrogen of the amide group, which is not very basic. It grabs a proton from some acid source, if you're following the acidic pathway, and then the carbonyl carbon can be attacked by oxygen. That gives the carbonyl group back its OH, and then you kick out the amine group. And so you break them up. Basically, all you need to know is that it is a net addition of water across the bonds. So you give 2 hydrogens to nitrogen, 1 oxygen to the carbonyl, and it is catalyzed by the enzyme protease. Okay, that was topic number 2 of amino acids, peptide linkage and hydrolysis. Now, topic number 3 is protein structure. And I've sort of been alluding to protein structure this whole time, but why don't we address it and give it its own proper treatment? So, there are 4 levels to protein structure: primary, secondary, tertiary, and quaternary. When it comes to primary structure, we're talking about the peptide linkages between amino acids to form a chain. The primary structure of a protein is going to literally just be the chain of amino acids stuck together. Now, there are going to be 2 ends to every peptide chain. Because you make the amide bond in the middle, you still have the amine group on one side and the carbonyl carboxy group on the other side. So what you can call those 2 sides of the peptide chain is the N-terminus and the C-terminus. N for nitrogen of the amine group, and C for carboxylic acid of the carboxylic acid. Now, important to remember that the Milestone Reviewed sheet reminds us of is that when it comes to the amine group, it is going to be protonated, it is going to be positively charged at physiological pH. And when it comes to the carboxylic acid group, it is going to be negative charged, it is going to be deprotonated at physiological pH. Now, let's move one rung up the structure ladder. 2' structure. The 2' structure of the protein is the local structure of neighboring amino acids. And there are 2 things that will stabilize 2' structure. One is the hydrogen bonding between the amino group and the non-adjacent carboxyl groups. So what does this exactly mean? Well, suppose you have a carbonyl, right? C double bond to O. That is a hydrogen bond acceptor, right? It can accept the positive charge on a proton hanging off of another molecule, right? An intermolecular force. So the carbonyl oxygen is a hydrogen bond acceptor. Well, what might donate a hydrogen bond? Well, we need an electronegative atom bound to a hydrogen. That's exactly what we have in the amine nitrogen of an amino acid backbone. So a polypeptide backbone, rather. So in the 2' structure, you are going to see hydrogen bonding, which is a very strong intermolecular force, not a bond, but a very strong intermolecular force, between the carbonyl oxygen of one part of the chain and the amide nitrogen of another part of the chain. And it is also the amide oxygen. Really, we're talking about the same thing. We're talking about the double bound O, single bound N. Now, this can form two common types of 2' structures. It can form an alpha helix, which is a common 2' structure defined by clockwise coils around a central axis, or it can form a beta pleated sheet, which is a 2' structure characterized by rippled strands that can be parallel or anti-parallel to each other. Important to note here is that when it comes to forming your 2' structure, which is the very local folding of amino acids, you would want something like glycine in the kinks on the inside of the alpha helix, but you would not want something like proline, because proline can interrupt the 2' structure because of its rigid cyclic structure. Okay, so important to mention here, proline can interrupt the alpha helix. However, it is often found in the turns and the loops in the protein. So it is important for turning and looping, but it can also interrupt the 2' structure. So one situation I can imagine this being important in is when you are causing mutations. If you mutate something that is not causing a kink into proline, it is going to cause a kink, and then you're going to have an interruption in your 2' structure. All right, let's move one more rung up the structural ladder. So this is the 3' or tertiary structure. Now, as I mentioned before, there is one important amino acid and one important covalent bond that can form in the 3' structure. That is the disulfide bond. It is the bond between the two sulfurs of two cysteines, which forms cysteine when it's together. But the miles down sheet reads, the 3' structure is the 3D shape of a single polypeptide chain, and it is stabilized by hydrophobic interactions, right, between two hydrophobic groups keeping each other away, acid-base interactions, which is the salt bridge interactions between something like glutamic acid or aspartic acid and lysine or arginine that I described before. And then there is H bonds, right? So hydrogen bonds are always important. And then disulfide bonds. Disulfide bonds are the big guys of the 3' structure, and the 4' structure too. I mean, really, disulfide bonds are the only covalent interactions you're going to find here. The rest are just IMFs. Okay. Or salt bridge interactions, which aren't actually bonds between covalently bound atoms. Okay, so that is the 3' structure. So miles down actually expands a little bit on what hydrophobic means. So this means push hydrophobic R groups to the interior of a protein, which increases the entropy of the surrounding water molecules and creates a negative Gibbs free energy. Yeah, so that makes sense. You want the hydrophobic R groups on the middle, on the inside, and you are going to have that in the 3' structure. Interesting. So the 3' structure is where we really start to see the middle start to be hydrophobic and the outside start to be hydrophilic of the protein. Now, important to note here is that when we're talking 3' structure, we're still talking about a single polypeptide chain. That is not the case with the 4' structure. So, Miles Down defines 4' structure as the interaction between peptides and proteins that contain multiple subunits. So, many proteins are going to have multiple subunits that can combine to form larger units. And you would still call that larger thing a protein, but you need to consider the fact that they're formed via those subunits. So where do those subunits come when it comes to multiple, where do those subunits combine? They combine in the 4' structure of a protein. One example that I could think of that would be commonly mentioned would be that of hemoglobin. So hemoglobin is the oxygen-carrying protein within a red blood cell, and hemoglobin binds to oxygen through interactions of 4 subunits. I believe it's 4. But basically, what's important to mention here is that that is a quaternary structure of hemoglobin, right? The 4' level, when we're talking about those subunits, that is a quaternary structure. Those are not single polypeptide chains that bind to those oxygens. That just doesn't happen. So yeah, quaternary structure. Kaplan mentions that quaternary structure can be denatured via heating and solutes. So yeah, denaturation, that's also something that Miles Down mentions. The loss of 3D structure caused by heating or solute concentration if you have very high solute concentration. They also mention 2 other things called conjugated proteins and prosthetic groups. I'll just read out the definitions of them here since I haven't really covered it. Conjugated proteins are proteins with covalently attached molecules. Okay, and prosthetic groups are the attached molecule in a conjugated protein. So that could be a metal ion, a vitamin, a lipid, a carbohydrate, or a nucleic acid. So interestingly, I would think that is hemoglobin. Let's see, is hemoglobin. Yes, okay, so conjugated proteins are those with covalently attached molecules, so the protein literally has something else bound into it, because you need to maybe have something as part of the protein that's not necessarily just polypeptide chain. So that could be a metal ion, vitamin, lipid, carbohydrate, nucleic acid. I believe in the case of hemoglobin, it is a metal ion, which is iron. Okay, so that is so the prosthetic group is the heme group attached to the protein structure. So the protein component is so this is actually it's not just the iron. The heme group is the part that contains iron, but it is the heme group that is the non-protein part of hemoglobin. So Google reads, yes, hemoglobin is considered a conjugated protein because it consists of a protein component, globin, combined with a non-protein part called heme, which contains iron. This makes it a complex molecule with a prosthetic group, heme, attached to the protein structure. So if we were to unravel a hemoglobin, we would see the polypeptide chain only in the globin part, but not in the heme. The heme is the prosthetic group, the globin is the protein. And overall, we would call that a conjugated protein. Okay, so that is the third topic, protein structure. Number 4 is the acid-base properties of amino acids. And this is an important one, this is a big one, because when it comes to amino acids, we are going to want to talk about what happens when there are different pHs to the R groups. So why might that be important? Well, when you have a protein, you're going to have those R groups sticking out of the polypeptide chain. And those R groups can either accept or donate protons. So it is very important to realize where R groups are going to accept protons or they're going to give them, and where they, like histidine, are going to sort of be in the middle, which we would call a buffer in the context of acid-base chemistry. All right, so just to begin, let's just define some numerical terms when it comes to acid-base chemistry, just as sort of a review. K is a constant that relates the concentrations of products and reagents of any equilibrium chemical reaction. So K is equal to the concentration of products over the concentration of reagents. K little a, K subscript a, it refers to just any, it's just a K, but it refers specifically to acids. So the K of an acid is going to be the concentration of the product over the concentration of the reagents, and when it comes to an acid, the reagent is AH, prototypical A acid, H proton, and then the products there are going to be A minus and H plus. So the A loses its hydrogen, becomes an A minus, and the H becomes its own proton. So you could donate a proton to solution, which really means donating it to water to make hydronium. Okay, so in the case of water, water is an amphoteric species, which means it can be described as an acid or a base. But just to describe what the Ka of water is that it has its own name, which isn't typical of acids, but water is very special. So it gets its own name, which is Kw, W for water. And the Kw is equal to the concentration of hydroxide times the concentration of hydronium at equilibrium, which is, when you multiply those together, it is 10 to the negative 14. So the P of anything is the negative log of that thing. And in the case of acid-base chemistry, we don't really want to talk straight concentrations like 10 to the negative 14. We want to talk simple numbers like 14. So if you take the negative log of 10 to the negative 14, you get 14. So the pKa of water is 14. Kw equals 14. Okay, pKw is 14. But when it comes to amino acids, what we're going to be talking about mostly is pKa. Ka, meaning the concentration of conjugate base and proton over the concentration of protonated acid, and is going to be the negative log of that value. So the pKa identifies how acidic a functional group is. The lower the pKa, the more acidic. The higher the pKa, the more basic. And this is because A higher Ka represents a higher concentration of protons over the protonated acid form, so you have more protons in solution. So a higher Ka is going to be 10 to the negative, let's say 2, for example, or 10 to the negative 3, whereas a lower Ka is going to be something like 10 to the negative 14, right, where there's a very low concentration of the proton in solution when you deprotonate your acid. So when we take the negative log of those values, we actually see a greater numeric number when we take the negative log of something like 10 to the negative 14. So we'll have like a pKa of 14 if your Ka is 10 to the negative 14. But that is actually highly basic, right? Because 10 to the negative 14 is a much smaller number than 10 to the negative 2. But if your pKa is something like 2, then it's going to be really acidic. So that means that the Ka is 10 to the negative 2, which means you have a very high concentration of proton in solution. So very important simple acid-base concept to remember here. Low pH, low pKa means very acidic. High pH, high pKa means very basic. Okay, so when it comes to amino acids, there are a few pKa values that are very important to memorize. So as part of this discussion, topic 4 of amino acids, I'm going to describe the pKa of the backbones and side chains of all the amino acids and give a bit of context as to why this value is what it is. Okay, so let's talk first the carboxylic acid and the amine group on any amino acid, and this is going to go for every amino acid, literally every single one. Now, the pKa of carboxylic acid is 2. It varies depending on what the R group is, but it's 2. Carboxylic acid is 2. One easy way to remember this is that there's 2 oxygens, pKa is 2. Meaning, when it comes to deprotonating this, you're going to need a solution that is more basic than 2, a pH of 2. Okay, so the pKa of all those carboxy groups on the amino acids is 2. That is greater than the pKa of acetic acid, which is 4.8, because you have a inductive effect from the amine group nearby, and depending on the R group, you might have even more induction. So pKa of the carboxylic part is 2. Now, the pKa of the amine group is 10. That's not the pKa of the NH2, because you know that's not going to be deprotonated. It's the pKa of the NH3 version of the amine group. So the NH3 version, the pKa is 10, much less acidic than the carboxyl group, but still very important to know. pKa of the amine, 10. pKa of the carboxylic acid, 2. Now, let's go into side chains. We are only going to talk about the amino acids that have acid-baseable side chains. Those amino acids are 1. Arginine. The pKa of arginine's side chain is 12. That is the pKa of the protonated imine on that double bound to nitrogen carbon on arginine. The pKa of histidine's side chain is the pKa of the proton that is bound to the protonated NH group in that 5-membered NH-NH ring on histidine, and the pKa of that is 6. Very importantly, the pKa of histidine is close to physiological pH, which is 7.3-ish. Sorry. So physiological pH, apparently, I've just received word, is (7.35 to 7.4). Okay, so anyway, what I was talking about there is that histidine's side chain has a pK that is very, very close to physiological pH. Important. Lysine's side chain. So this is the pKa of the NH3 group at the end of the butyl alkyl chain on lysine. That pKa is 10.67. 10.67. Glutamic acid has a very acidic glutamic acid and aspartic acid, right? These are the negative guys, the negative amino acids, because they get deprotonated very quickly. They're also the acidic amino acid. I meant to say acidic. So aspartic acid, and the conjugate base is aspartate. Glutamic acid, the conjugate base is glutamate. The pK of aspartic acid is 4, and the pK of glutamic acid is 4. So similar to carboxylic acid, it's 4. Now, let's get into, okay, so we're skipping serine, threonine, asparagine, glutamine, cysteine, glycine, proline. None of those pKs matter because this is the pK of the backbone. And that's it. So the pKs that are important to memorize for amino acids is 4 for the carboxylic acid ones, aspartic acid and glutamic acid, 10 for 10.6 rather, so really more like 11 for lysine, which is the NH3 group's ability to be deprotonated, 6 for histidine, and 12 for arginine. Now, what do we do when we are given a pH of a solution and we have to determine whether or not our molecule is protonated? Well, one thing that Leah mentions, which is an interesting way to think about it, is the fight for the proton. Is the pKa higher or is the pH higher? Whichever one is higher, that's the one that gets the proton. If it's the solution, then the pH is higher and it gets the proton, it gets the H+. If it's the molecule that has the higher value for its pKa than the pH of the solution, it gets the proton. So let's give an example here. Say you have histidine and you have a pH 10 solution. In that case, the solution gets the proton, histidine is deprotonated. Not negative, because histidine's nitrogen would actually be neutral when deprotonated, but since that pKa is 6, if you have a pH that's higher, the solution gets H and histidine is deprotonated. So pH wins the fight. Well, what if we have histidine in a pH 2 solution, a very acidic solution? Well, in that case, pH is lower than the pKa, the molecule wins the fight, and the histidine gets its proton for a positive charge. All right, so I've started throwing out these positive and negative charges, and in this case, it is now going to be very important to talk about the net charge of an amino acid in a solution of a given pH. So, when the pH is given to you, you may have to figure out, what is the net charge of one amino acid or a group of amino acids at that given pH. So Leah gave the example of glycine, so non-peptide bonded glycine. Now, glycine is a great example for this because it is only the backbone of an amino acid. It's only an amine group and a carboxylic acid group with an alpha carbon in the middle with 2 hydrogens, achiral. So glycine is going to display different charges depending on the pH of the solution. And we're going to go one by one to sort of explain here why those charges are what they are. Now, the pK of glycine's amine group is 10, the pK of glycine's carboxylic acid is 2. And when you have a pH of 1, that carboxylic acid wins the fight for the proton, meaning its pKa is higher than 1, so it gets its proton, and the NH2 also wins the fight because its pKa is 10, higher than the pH. So both of those win the fight. They both get protonated. That means NH2 is positive, and the carboxylic acid is neutral. The overall charge of this solution is going to be positive 1. Now, let's say we bump the pH to just above the pKa of the carboxylic acid group, 2.3. Well, it's 2.3, so it's not really that far above the pKa of the carboxylic acid group, so we're actually going to have a half and half mixture, for all intents and purposes, of the carboxylic acid protonated and deprotonated. Now, a half and half mixture of a carboxylic acid, protonated and deprotonated, is half positive and half negative, meaning the overall charge of that is negative 1/2. But the NH2 group is going to be protonated in every case because its pK is greater than the pH, meaning that it gets the proton, so it's protonated. Well, that means that the overall charge is going to be the positive 1 on the NH3 group minus the negative 1/2 on the carboxylic acid group for an overall charge of positive 1/2. So when we raise the pH, we went from a molecule charge of positive 1 to positive 1/2. Now let's go to pH of 7. At pH of 7, it is physiological pH. Remember, physiological pH is 7.35, 7.75 to 7.45, so 7.35 to 7.45, and at that pH, the carboxylic acid is protonated and the amine group is also protonated because sorry, carboxylic acid is not protonated, the amine group is protonated. You have the Zwitter ion form, which is net neutral, and so the solution is neutral. Okay, then why don't we bump up the pH to just around the pKa of the amine group, so you can see where this is going. You're going to get another half charge sort of thing. So at this point, the carboxyl group is definitely negative because, it's lost the fight. Its pKa is way lower than the pH. It's lost the fight for the proton. So we have a -1 from that, but then we are going to have a positive 1/2 from the fact that half of the amines are protonated, half of them are deprotonated, because the pKa of the amine group is 10. Okay, so now we bump it all the way up to a pH of 13, and we get a charge of -1 because the carboxylic acid is all deprotonated, and now at this very, very basic pH, finally, the pH has won the fight between pH and pKa for the amine group, and it is deprotonated. So the amine group is neutral, the carboxylic acid group is -1, and overall, it is going to be a -1 charge at a very high pH. Now, that's all well and good when we're determining the total charge and the protonation or deprotonation states of the functional group of a molecule at a given pH, which is an important thing to do, but we're going to be talking polypeptide chains when it comes to amino acids. So we're going to want to figure out how to quickly determine what the charge of the polypeptide chain is, and we're also going to want to determine the, at a given pH rather, but we're also going to determine the pH at which the polypeptide chain's charge is going to cancel out. So the polypeptide chain's overall charge is 0. And it turns out there's a special name for that point, which is the pi, also known as the isoelectric point. So this is the point, the pH at which the amino acid is in its Zwitter ion form. The charges cancel out to make a neutral molecule. So you can consider this for an amino acid like glycine, where it only has 2 functional groups and no polypeptide chain, or you could consider an entire polypeptide chain, where you're going to have those amide bonds between amino acid backbones, and you're going to have an N-terminus amine and a C-terminus carboxylic acid. So Leah gave the example of a polypeptide chain consisting of cysteine, lysine, and tryptophan. Now, when I was going through and listing out those pKa's, I touched on lysine. Lysine's pKa is 10.67, which is close enough to 11. So let's say that's 11. And then Leah also uses cysteine, which has a pKa of 8.1. So like an alcohol, that thiol group can be deprotonated, and that has a pKa of 8. Okay. So cysteines is 8, lysines is 11. Now, tryptophan, no ionizable groups here, X, so not applicable. The C-terminus is just a carboxy group, so that's 2, and the N-terminus is just an amine, that's 10. So the 10 and 2 are going to be constant for the C-terminus and N-terminus of any polypeptide chain. And then we have to determine in the middle, what are the pKa's of the ionizable groups that are coming off of those amino acids. For cysteine it's 11, for cysteine it's 8, for lysine it's 11. Now, we have those pKa's memorized, we write them out. Then we put them in order from lowest to highest. So the lowest here is 2 for the carboxy group, 8 is the second lowest for the cysteine, and then higher than that is 10 for the amine group, and then 11 for the lysine group. Now, what we want to do now is protonate all of the possible protonatable groups. So we've already drawn out the side chains for those amino acids, and particularly just the part that could be protonated or deprotonated. So for cysteine, that's SH. For lysine, that's NH2. Or NH2. Well, now let's protonate all of it. So that's going to be, for the amine group, NH3. For cysteine, SH, because that's protonated, cysteine, neutral. For lysine, it's going to be NH3 as well. For tryptophan, Na. And for carboxylic acid, that is going to be a protonated carboxylic acid. So you notice here that the cysteine and the carboxylic acid are neutrally charged, the tryptophan is not applicable, and the lysine and the amine group are both positive, which means the overall charge of this polypeptide chain is going to be positive 2, 1 for the amine, 1 for the lysine. Now that we protonated all the ionizable groups, we have our max Q, max charge. Q is just the physics term for charge. So, max charge. We want to start from the left and go to the right. So we've listed out our pKa's from lowest to highest, and we start from the left. So below a pH of 2, all groups are going to be protonated, and we're going to have the max Q, which is positive 2. Now, 1 to the right, and we have between 2 and 8, pH of 2 and pH of 8. And in this case, one of our groups is finally deprotonated. That's a carboxy group. So that one is going to lose its proton, and that removes one charge point from the overall charge. So that's going to be positive 1. Now between a pH of 8 and 10, you have zero charge, Zwitter ion, and then between 10 and 11 you have negative 1, and above 11 you have negative 2. So you can always go between these pKa values of the individual functional groups on your polypeptide chain and say it goes 1 charge point between each one, depending on what the pH is. So pH of 1, you're going to have positive 2, pH of 4, you're going to have positive 1, pH of 9, for example, you're going to have 0, and then pH between 10 and 11, you're going to have negative 1, pH above 11, you're going to have negative 2. Now, how do we find the pi, or isoelectric point, given the pH values that we just described and the resulting charges? Well, I said that there was a charge of 0 between pH of 8 and pH of 10. That means that we can find the isoelectric point by simply taking the average of 8 and 10, which is 9. So it turns out that you can do this with many different amino acid functional groups. So finding the isoelectric point is going to be just taking an average of 2 pKa's and dividing that by 2. So if the pKa of one is 3 and the pKa of the other, or if the pKa of one is 2 and the pKa of the other is 4, you divide that by 2, and you get 3 rather. Okay, so be careful with the math there. But the steps here are writing out the polypeptide chain with the functional groups, ionizing all of them to find Qmax, ordering out your pKa values, and then starting from Qmax and going 1 down for each jump between pKa values that you listed out. So the milestone review sheet sort of puts this into context for each different amino acid chain, and so it gives a formula to find, an equation rather, to find the pi of any sort of side chain. So if you don't have any side chain, then you're just considering the carboxylic acid and the NH3, which have pKa's of 2 and 10 respectively. So the equation for pi here is going to just be the 2 pKa's divided by 2, right? So we have the 2 pKa's, and it's going to be Zwitter ion somewhere in the middle. So we have COOH and NH3. So it's going to be COOH plus the NH3 divided by 2. And when I say those functional groups, I mean the pKa's of those respective functional groups. For a neutral side chain, such as alanine, that's going to be the same equation, because all you have is the carboxy group and the amine group. Now, for a basic side chain, you're going to take your amine group, but then you're also going to take your R group, because we're dealing with basic here. You're going to divide those pKa's by 2. So for lysine, for example, its side chain has a pKa of 11. That's a pKa of 11 because it is an NH3 group, and in order to find the pi of that, you're going to take that NH3 group, add it to the NH3 group on the N-terminus, and divide that whole thing by 2 to find the pi. Now, to find the pi of an acidic, you do the same thing, except the other pKa that you're adding, the 2 pKa's that you're adding are that of the carboxylic acid, which is 2, and then that of whatever the functional group is. So, for example, for asparagine, that would be like 4 for its functional group. Okay, so I'm going to just touch briefly on titration here. So the midpoint of a titration is where the pH equals pKa. The equivalence point of a titration is where the pH equals the pi. Very important. Okay, so we've covered section number 4, which is acid-base chemistry. Now let's talk about number 5, stereochemistry. So stereochemistry is typically referred in organic chemistry to a discussion of R versus S, but in fact, we use a different convention in biology and biochemistry. So that is the convention of D versus L. So D and L has nothing to do with R or S, but it does have something to do with the side on which the carbonyl group when it comes to sugars, or the amine group when it comes to amino acids, is on the Fisher projection. So the Fisher projection is just like flattening out your molecule and having a patchwork of bonds which are linearly arranged. And the D form is going to have for amino acids, we'll talk about sugars a different time. For amino acids, the D form is going to have the amine group on the right, and for the L form, the amino acid is going to have the amine group on the left. One can remember this by remember L, left, right, droite. Okay, so eukaryotes tend to like the L version of amino acids. And really, the stereochemistry, we say there's no equivalence between L and D and R and S, but that's not true. For every amino acid, the L version is the S absolute configuration. For every amino acid, the D version is the same as the R configuration. There's one big exception to this, which is cysteine. So for cysteine, it's going to be opposite. The D is going to be S, and the L is going to be R. But there's a good mnemonic you can use to remember that any amino acid is going to be D. The D form is going to be the R absolute configuration. The L form is going to be the S absolute configuration, and that is using doctor. So doctor is an abbreviation, is abbreviated by DR, dr dot. So the D in doctor is for D, and the R for doctor is for rectus, which is one of the two absolute configurations, right, when we're talking about S versus R. So DR, D equals R. So if the D is R, the L must be S. So that's a really helpful way to sort of remember your D versus your L and your S versus your R. I'm not 100% sure how that is going to be tested or important, but, it's a thing to mention here. Okay, so just a quick recap of everything we've covered in this discussion, quite long. We started by mentioning just what an amino acid is and why we are only talking about the 20 proteinogenic ones, and then we went into the categories, that is hydrophobic, which encompasses aliphatic and aromatic, hydrophilic, which encompasses just polar and then charged side chains, and within those, there are some special amino acid properties that are important to remember. In the second topic, we talked about peptide linkages, which is the condensation or dehydration reaction that forms the bond between the carbonyl carbon and the amine nitrogen of amino acids. And then we also talked about hydrolysis, which is just doing the opposite, adding water across that bond and breaking it up. And we talked about the respective enzymes that do that. So for peptide linkage, for example, that's like ribozyme, right, because it's in the ribosome. I forget which, the protease, I guess, is the one that splits them up. Okay, whatever. So number 3 was protein structure, which is, 1 prime, 2 prime, 3 prime, 4 prime. Remember, the only covalent bonds on the 3 prime structure is the disulfide bonds between cysteine amino acids, which form cysteine. Number 4 topic that we covered was the acid-base chemistry of amino acids. So we covered the pH, the pKa, and the pi, which is the isoelectric point, the pH at which the overall charge of the amino acid is going to be neutral. And then we also covered topic number 5, which was stereochemistry, which is the D and L and D and L and the R and S. So D equals R, L equals S, except for cysteine. Cysteine is the special case. All right, so that covers the first important biochemistry topic on the MCAT, which is amino acids.